Opsis has been a little bit under the radar. It's formed about 3 years ago, ideation and development about 3 years prior to that. And so, I've had people ask me what the company's about. A relative texted me asking me what it was about, and I had texted him back that it's about curing certain forms of blindness. And my phone auto-corrected that to certain forms of blondness, and so I'm in favor of curing one of those. Where's the, is this the, okay. So the company, as I mentioned, was formed in 2016. It's based in Madison, Wisconsin. It's built on top of the platform technology that CDI, later acquired by Fujifilm, had developed, which is where I came out of. The company is developing advanced medicines for patients suffering from inherited retinal disorders, or IRDs, which includes retinitis pigmentosa broadly, and a number of the indications that were discussed here. As well as really the large target would be AMD. We're focused not on genetic repair, but on anatomical repair. And so I think that we complement the success of the gene therapy and gene editing industry, where they might be able to intervene early in specific areas, and we intervene later in patients that have degenerated past the point where you can expect a palliative or a gene editing therapy to have an effect. So the team includes Nick, who gave a presentation earlier. Dave Gamm is the co-founder and really the scientific founder of the company, and it's based on science from his lab as well as from a number of our scientific advisory board members like Kapil Bharti at NEI, for example. From the R&D side, Lucas Chase heads up our development along with Joe Phillips on the preclinical, and so these are people who have been in the space for a long time developing retinal and or CNS-based iPS cell products and therapeutic products in development, and a lot of experience with the preclinical animal models that are necessary to evaluate this. So our view is that, again, to complement what the successes are in the gene editing and gene therapy space, there needs to be a strategy for cell repair and cell replacement. There need to be replacement parts for the retina. There's going to be a large number of patients on top of the ones that already exist. If you look at the spread of inherited retinal disorders and the genetic causes of them, it's very broad. And a large swath of that is, as of yet, uncharacterized. And so while it makes sense to be opportunistic about intervening early in disease where you can and where it mechanistically makes sense, it also is necessary to be able to address kind of the back end or the larger tail end of this problem, which is how do we help repair people who have anatomical related vision loss. And so the foundational science and technology was really based on pairing up the proof of concept on authenticity of a photoreceptor and retinal cell lineage development developed by the Gamm lab, Dave Gamm, as well as many other people in the field had developed this type of science. And so this is part of our foundational science. What we did in forming the company was to marry that with the industry-specific CNS IPS scale-up experience that came out of the CDI, then Fujifilm platform. And so the basis of this was, Dave Gamm and his team identified that you could grow optic vesicles in vitro, and that these vesicles would harbor a laminar retinal layer that's very similar to what you see in the eye, and that this is a difficult process to scale. So it's an organoid, you're kind of picking things out. It can be a challenge to scale it. And so we took this authenticity again and paired it with the ability to scale it. And instead of presenting a pipeline, I guess what I was interested to just put up here was what we view as the retinal cell platform. Starting from the retinal progenitor, there are a number of different lineages you could make or focus on. We focused on what's shown here initially as the RPE product, which a lot of groups are also working on. Our calling card is really to develop this photoreceptor, what we call PRP product, and then to take the two products and produce a combined graft for AMD, for example. And so each of these has a place in the patient population, but we put it up here to illustrate that this is the purview of our expertise and our technology platform. And so in the future, for example, it may make sense to make rotor cone-specific products. It may make sense to make retinal ganglion products. and this is part of the way we're thinking about things. Photoreceptor Product took us about 3 years to conduct lead optimization around and to develop the right method to find the Goldilocks zone in which we would have a scalable process wherein we're producing cells that bear the authenticity that, for example, Dr. Gam had developed. And so it's an iPS-derived program. It takes a significant amount of time to produce the target cell, shown here around 70 days. But it's very robust, and we can produce large quantities of these cells at high purity, and we can cryopreserve them. And so that's our objective in producing this, what we believe has the potential to be a first-in-class product. And I won't go into the details of the process itself, but what I'll say is that when you start to look at purity and characterization, and there were a number of different candidates we evaluated in vivo in the animal models that we're looking at, it became increasingly important for us for this specific lineage to ensure that you don't have off-target cells. And I think that the need to rid your product of off-target cells varies depending on what you're making. But in this particular case, if you're in the business of making photoreceptors and you're putting them in subretinally, you must avoid certain things like proliferative progenitors, for example. And so where we've landed is, again, a very robust way of making very high purity cells, shown here in this single-cell-seq data that, we're getting high levels, for example, of recombinant positive cells routinely in terms of our flow sort markers. It's a very high purity product. And so that's the lead candidate that we are taking forward. Lead op, we made a conscious decision at the outset of the company during lead op that while there are many animal models in the retinal space to consider, a lot of them are potentially confounding. And so the question becomes, do you want to satisfy the safety requirements and do the experiment in human? Or do you want to have a really good sense for what you can learn in the animal model realistically as it relates to the limitations of these retinal models? And so we chose to do our lead op in a retinal degenerative model called S334-TER, which has a fairly extensive degeneration of the photoreceptor and outer nuclear layer. And so bearing that in mind, the purpose of this model wasn't to show functional recovery, which is very challenging, but rather to show that we could, in a stepwise way, verify the anatomy we're looking to recapitulate in the clinical trials. And so that's been our focus. And what we've been able to do over a number of cohorts of animal studies is show that we can rise to the level where the vast majority of the cells we transplant survive and are maintained stably in the grafts and then begin to mature. And so that is really what our objective was. This is a plot, just the data on the right is at 3 months, but another factor is, where are the cells delivered? So you produce the bleb in the subretinal procedure, and then is this something where you're going to put the cells in and they're going to stay in one area and clump, or are they going to be able to cover the area of the bleb at least in which you're looking to alleviate the anatomical defect? And the method in which we're formulating the cells and delivering them, the delivery is fairly standard for the photoreceptor product through an off-the-shelf cannula. And as it happens, it's very robust in terms of getting very even distribution, as shown here, just to illustrate that we're not showing data that's the cherry-picked region of the animal model wherein we find a positive engraftment, but rather it's fairly uniform. Not only is it surviving very well, but it's a fairly uniform graft as we're producing, at least in the animals. And then the key thing is, what do the cells do once they get there? We're putting in progenitors. We tested, of course, more mature or less mature cells and found the candidate that we saw the best results with the least off-target effects. And that is, showing that we can observe maturation of the cells into both rod and coned, about a 50-50 mix of rod and cones in this particular candidate in development. And so that was a significant validation for us of our thesis in terms of what product to develop as a lead candidate. The next question really then becomes, can they integrate? So do they start expressing the synaptic machinery? Do you see, for example, in the right shown here with PKC alpha staining, do you see the host bipolar infrastructure start to innervate the graft? And in fact, it does. And so again, while we're not after necessarily a really black and white functional recovery, given the differences between human and rodent synapse, for example, what we're looking for is we want to be confident before going into human that A, it's safe, of course, but B, that it has a reasonably good chance of having a high effect size. And so that's our objective. In terms of further maturation, if you start to go out 6-9 months, then you begin to see what was our goal, but is nonetheless amazing to us, which is the robust formation across the grafts of outer segments of both rod and cone cells, as I mentioned earlier. You see a number of other components, of which this is only a small subset, but you're also seeing other signals like correct trafficking of rhodopsin, for example. And so these persist long-term and they continue to mature. And so it suggests to us a couple of things in terms of the potential of the technology to help humans that have these diseases, but it also provides us with a pretty robust understanding of what we should expect in the clinic. If we do transplants, a few years from now, and observe an immediate recovery of some kind, it doesn't match up with what we know in terms of the anatomical process in the animals, and so that would confuse us, and we might think that that's not really a real effect. Whereas if it sort of follows the developmental paradigm that we're seeing from the anatomy, then that's a signal to us that the mechanism that we purport is in fact correct. So those were small animal data. Of course, we're looking in large animal as well. Again, not so much for sort of functional recovery, just because of the fundamental differences between human and other mammalian, not only the eyes, but the synaptic machinery as it relates to retinal transmission. But we want to practice our surgical technique in the large animal. We want to validate that the grafts do what they do in the small animal, which is to survive, innervate, et cetera. So this is data from a P23H rhodopsin mutant in partnership with NEI, and so we're seeing similar results that we see in the rodent. And that's very encouraging. We also have ongoing studies with the group at UPenn in the canine models. So this is ongoing, and I think supportive of our argument for potential potency. So that was all about the photoreceptor product, which again is kind of the quote-unquote simpler case where you can deliver a high effect size product potentially in using an off-the-shelf delivery approach. With retinal pigment epithelium delivery and repair, or as what we're really interested in ultimately is a product that has retinal pigment epithelium and photoreceptor as part of an integrated graft. That's our really big long-term objective. That's a harder problem, both in terms of delivery and the optimization of those types of grafts. And so we've been working on that, of course, in parallel, and we're doing it in partnership again with our collaborators at the National Eye Institute, building upon the techniques and technology that we've co-developed with them over the last several years. And so this is just some data illustrating that a hard problem, historically has been how do you get photoreceptors to adhere to the RPE? And how do you do that in a way where you can still deliver it as an intact graft? And so I think we've made a lot of progress towards that. And on the right, those are just showing some in vitro imaging, and on the left, some further imaging. But these grafts are similarly also going into large animal studies now. So that is everything for us. So I'm happy to talk to people or take questions.